Today we are going to study about enzyme nomenclature and classification. Enzymes were commonly named by appending suffix "ase" to the name of enzyme substrate. This system was given by Ducklogs. For example, arginase, which acted on arginine. Another example is urease, which acted on urea. This practice resulted in anomaly because two enzymes were given the same name or vice versa. Two different names were given to the same enzyme. Therefore, this system was not followed. Many enzymes were given names that provided no clue to their function. For example, catalase, which acted on hydrogen peroxide. So this system was also not followed. Later, enzymes were classified by International Union of Biochemistry. IUB recommends that the overall reaction as expressed by formal equation be taken as the basis for naming enzymes because the chemical reaction catalyzed by the enzyme is its specific property which distinguishes one enzyme from another. This system was used for classifying enzymes. Although this system is complicated, it is precise, descriptive and informative. It is complicated because the whole equation is used for naming the enzyme. It is precise because each enzyme has a specific name, and it is descriptive because it tells you everything about the reaction. It is informative because it tells us which reactants are reacting, what kind of reaction is taking place. Therefore, it helps us to distinguish one enzyme from another. IUB devised a system for assigning code numbers to enzymes based on classification. Code numbers are used with the prefix EC. Each enzyme number contains 4 elements separated by points, and they are arranged on these principles. Enzymes are classified into 6 main classes on the basis of chemical reaction they catalyze. Class 1, Oxidoreductases. Class 2, Transferases. Class 3, Hydrolases. Class 4, Lyases. Class 5, Isomerases. Class 6, Ligases. Each class has a specific number, and this number cannot be changed. Each class is further subclassified into several subclasses on the basis of bond splitted or formed, group transferred, or by classification of the general reaction type. Subclass further describes the subclass. So, the first figure tells us about the class, second figure about subclass, and the third figure tells us about subclass. The serial number of the enzyme in its own subclass is indicated by fourth figure. The systematic name includes the name of substrate or substrates in full and a word ending in ase indicating the nature of process catalyzed. The classification and assignment of EC numbers. Class 1, Oxidoreductases. They are enzymes which are involved in biological oxidation and reduction reactions. These enzymes catalyze transfer of hydrogen atom, oxygen atom or electron from one substrate to another. General reaction is written as X oxidized plus Y reduced will give you X reduced plus Y oxidized. Second figure represents subclass. It tells us about donor substrate undergoing oxidation. It means which substrate is donating hydrogen or electrons. If subclass is 1, then alcohol is the hydrogen or alcohol is the hydrogen or electron donor. If subclass is 2, then aldehyde or ketone group is the hydrogen or electron donor. Third figure represents the subclass. For class 1, it shows the type of hydrogen or electron acceptor. That means which substrate is accepting hydrogen or electrons. If subclass is 1, then NAD plus or NADP plus is accepting hydrogen or electron. If subclass is 2, then ferric ions, for example as in cytochrome, are accepting electrons. The systematic name of enzyme is based on donor acceptor oxidoreductase. For example, class 1 oxidoreductase. 1.1 acting on CHOH group of electron donors, that is oxidoreductases acting on CHOH group of electron donors. 1.1.1, that means oxidoreductase acting on CHOH group of electron donors with NAD plus or NADP plus as acceptor. Specific example, EC 1.1.1.1 is alcohol NAD plus oxidoreductase. Trivial name is alcohol dehydrogenase. Class 2, transferases. These enzymes catalyze those reactions in which a group X is transferred from one substrate to other. The reaction is AX plus B will give you BX plus A. Here the enzymes transfer amino group, methyl group, glycosyl group, etc. from one compound regarded as donor to another compound regarded as acceptor. The systematic names are formed according to the scheme donor, acceptor, group, transferase. The subclass here denotes the group transferred. If subclass is 1, then one carbon group is being transferred. If subclass is 2, aldehyde or ketone group is transferred. If subclass is 7, phosphate group is transferred. The subclass further describes the nature of group transferred in subclass. If EC is 2.1.1, then the enzymes are methyl transferases. Here, this denotes that the subclass 1, one carbon group is being transferred, and subclass is also 1, then one carbon group is methyl group. Another example is EC 2.7.1. Enzymes are phosphotransphorases with alcohol group as acceptor. Here the phosphate group cannot be further described, therefore the acceptor is mentioned in subclass. Hence transferases are enzymes which catalyze transfer of a group other than hydrogen between a pair of substrates. For example, EC 2.7.1.1 is ATP dehexose 6-phosphotransferase. Trivial name hexokinase. The reaction is ATP plus dehexose will give you ADP plus dehexose 6-phosphate.